So I'd like to welcome you to this satellite symposium on immunotherapy in maintaining remissions in AML. And I'm extremely thankful to Immune Pharmaceuticals to giving us the opportunity to discuss this very interesting topic. As you may have seen in my previous presentation, I consider immunotherapy an extremely challenging approach in AML, which is really not very well investigated so far, except for a new agent that we are discussing, and which for some reason, which I really don't understand, is not really in our main focus. So hopefully this will change by this satellite and the two presentations that we are looking forward. And the first presentation will be given by Professor Hellstrand.
Thank you very much, and thank you for the kind invitation here, and thanks also again to Immune Pharmaceuticals for supporting this symposium or this presentation. My talk will revolve around histamine dihydrochloride for use as a relapse preventive immunotherapy in AML, and I will focus on certain pharmacological aspects of this therapy, immunomodulation based on inhibition of the NOX2 enzyme. This work was much of the work that I will present has been conducted at the University of Gothenburg, and preclinical and translational results have been gathered by Drs. Fredrik Berg-Thorén and Anna Mattner at that university. There you go. I'd like to introduce you to NOX2. NOX2 is the myeloid NADPH oxidase. It's almost exclusively expressed by myeloid cells, monocytes, and granulocytes, and it is indeed expressed also by certain leukemic cells in AML, the FAB4 and FAB-M4 and M5 classes, along with CML cells and CMML cells that almost invariably express the NOX2 enzyme. This enzyme takes up molecular oxygen and converts it into toxic reactive oxygen species. It begins by the superoxide anion and then converted into a whole family of oxygen radicals, as they are popularly referred to. Once released into the extracellular space, these agents, the ROS, will participate in antimicrobial defense, but they are also strongly immunosuppressive, in particular towards two types of cells, and that is the natural killer cells, the NK cells, and subsets of cytotoxic T cells. And I will show you some examples of that, beginning with the NK cells. You can see from this slide that NOX2 positive monocytes, human healthy monocytes, CD14 positive cells that express almost invariably NOX2, very efficaciously inactivate NK cells. The upper part of the slide is human sorted NK cells at a very high purity, and the lower part is the same cells incubated with monocytes that release ROS to inactivate NK cells by apoptosis, signified by the yellow color. If you do this experiment in the presence of Ceplene or histamine, you will see that much of this apoptosis is completely prevented, and that is the action of Ceplene to inhibit the formation of immunosuppressive ROS. We can see that this is not restricted to NK cells, human T cells in particular. The T effector cells that are assumed to exert anti-leukemic action in AML are equally protected by histamine. You will see from the first picture that we have a population from this FACS plot of viable sorted CD8 positive T effector cells. If you add in monocytes, again NOX2 positive monocytes, they will release radicals to inactivate the T cells by apoptosis. The green population of cells is the apoptotic T cells, and this is almost completely prevented in the presence of histamine. And this is a summary of similar experiments done in cytotoxic T cells, where histamine, as it seems, almost completely prevents oxygen radical-induced apoptosis. Now, another feature of histamine is that it synergizes with immune activators such as IL-2 to improve their efficacy. On this slide, on the upper part, you see an experiment. I think that many of you may have done similar experiments where you activate human NK cells by IL-2. They will, within a few hours, acquire cytotoxic activity, but also express the activation antigen, CD69, signified by the yellow dots. If you do the same experiment in the presence of NOX2 positive monocytes, you will see very little activation of the NK cells by IL-2, whereas if you include histamine along with IL-2, you will restore this activation. When these were experiments that actually were published several years ago, and we were interested in see whether this action of histamine together with IL-2 translated into anti-leukemic efficacy in vitro, and we did experiments using AML cells from humans as effector cells in microcytotoxicity assays. What you see here is you see here NK cells incubated with labeled AML blasts, and we measure the killing of the AML blasts by NK cells. What you see, first of all, is that there is very little constitutive cytotoxic activity by NK cells against AML blasts, whereas in these experiments, more than 50% of the AML blasts were indeed killed by IL-2 activated NK cells, emphasizing that IL-2 is endowed with significant anti-leukemic efficacy. Now, if you did the same experiment where you mingled in or mixed in NOX2 positive monocytes, you can see that much of this IL-2 induced activation was absent and again restored by the presence of histamine or Ceplene. So we were encouraged to combine these two agents for, as we believed, optimal anti-leukemic efficacy. I will not go into the many animal experiments that we did with this combination of drug with histamine as an anti-tumor agent as monotherapy, and in particular improving the efficacy of IL-2. We'll instead move directly to the proposed anti-leukemic mechanism of histamine. I've made this animation where you have a myeloid cell here. On most of my slides, it's a monocyte. That the important thing is that it expresses the immunosuppressive NOX2 and an anti-leukemic effector cell in the middle, and these cells are AML blasts. What you see first is that the myeloid cell will take up molecular oxygen and convert it into toxic reactive oxygen species that are very toxic to NK cells, which has important functional consequences. You will see that activating receptors, for instance, on these cells will be reduced. And as you have seen from the previous slides, a substantial of these cells will lose their cytotoxicity and will not respond to IL-2. And eventually, NK cells will acquire apoptosis by this interaction. I will now introduce Ceplene. I will have to warn you because it comes in very rapidly on this animation. This is the H2 receptor, the histamine H2 receptor that Ceplene acts by. It comes in from the left now to target this receptor and inhibit functions of NOX2. And the consequences are that oxygen radicals are no longer produced, and the functional consequences, as you again have seen, is that there is no apoptosis. The anti-leukemic effector cells remain viable. If I may draw your attention to the arrows up here, you will see that the activating receptors are maintained and the cytotoxicity of the NK cells is maintained. These cells will respond to IL-2 and lyse AML blasts. Then we started in 1997 or 98 immunotherapy for AML patients in remission. And you may ask, why did we opt for AML? Well, there are many reasons for that. Of course, we know that immunotherapy in a broader sense is efficacious in AML. Experience from allotransplantation is quite clear, and there is certainly evidence to suggest an anti-leukemic function of NK cells in transplanted patients, but also in patients who are not eligible for transplantation. And perhaps even more importantly, there is a very small or minimal clone of malignant cells that is dangerous in AML after the completion of induction and consolidation therapy. I have tried to illustrate that with this slide. This is at least an illustration how few cells that can be even, that will eventually expand and have severe health consequences to a patient with AML. We did clinical trials on the basis that IL-2 was supposed, as I've mentioned, to activate optimally anti-leukemic functions of lymphocytes, and that histamine would protect these lymphocytes from inactivation, ideally to achieve destruction of leukemic cells and prevention of relapse. We started, I will very briefly go through some of the clinical experience, with phase 2/1/2 trials in Sweden, especially in Gothenburg and at the Karolinska Institute. We observed low or acceptable toxicity with minimal impact of quality of life in treated patients and decided to proceed to phase 3. And I will introduce you to the phase 3 trial that was completed in 2005 with 320 patients with a primary endpoint of leukemia-free survival at 3 years. Patients took sub-Q injections of Ceplene and IL-2 as home treatment during this trial. This is a summary of the phase 3 trial results. We met the primary endpoint of LFS in all patients, but the treatment effect seemed to be more pronounced in those 261 patients that were in first remission at onset of therapy. And within that group, there was a striking effect in younger patients. After that, when I was here last in 2011, we had asked what subtypes of AML is perhaps, where is the treatment most beneficial? And the answer was, as it seems from post-hoc analysis, monocytic forms of AML, the treatment seems to be more efficacious in M4 and M5 Fab classes of AML, and the reason is arguably that these subsets of AML comprise leukemic cells that express histamine receptors and NOX2. I'll have to refer to this paper where we describe this in more detail. So there is a preferential activity of this combination, histamine and IL-2, in monocytic forms of AML. You see on the left-hand side M2 AML, where we almost never can detect receptors for Ceplene or NOX2 on the cells of the leukemic clone. And in Fab M4 leukemias, this is patients in CR1 below the age of 60, where there is a, as it seems, pronounced treatment effect. If you do the calculation of excluding the Fab M2 AMLs, the treatment effect is even more pronounced. I'd like to emphasize that this is post-hoc data and need to be confirmed in subsequent trials. Now, we have just recently completed a phase 4 trial, and in this phase 4 trial, it's called the Remission phase 4 trial. We included 84 patients to study a variety of aspects of AML, including cellular immunity and biomarkers of efficacy. And for this particular presentation, I will show you data on immunity. We asked, does histamine and IL-2 therapy indeed activate NK cells as predicted from the preclinical experience? And does it impact on the function of the cytotoxic T cells? And finally, do these aspects of immune function translate into clinical efficacy in terms of relapse risk or relapse prevention and overall survival? I'd like to begin by showing an outline of the trial. And patients were slightly dominated by older patients, a median age of 63, 47 of whom were above the age of 60. Patients were included after completing consolidation therapy and were scheduled for 10 3-week cycles of Ceplene and IL-2 with intermissions as outlined here. And I'll show you some data again on immunoregulation and begin by the regulatory T cells. I'm sure that many of you know that IL-2, one of the components of our regimen, is an inducer of regulatory T cells, which, on the other hand, have been ascribed a negative role for immunity, and that seemed to accumulate in certain forms of cancer. We observed a striking induction of regulatory T cells during each cycle of Ceplene IL-2. On the upper right, you see a FACS plot where patients start with approximately 3% of the CD4 positive cells in blood being regulatory T cells, and even already at the end of a treatment cycle 21 days later, the regulatory T cells become the dominant population of CD4 positive cells in blood. And on the right-hand side, you see that this is repeated again in cycle 3, found in cycle 1, but repeated again in cycle 3. Somewhat to our surprise, I would like to mention that we sorted out these regulatory T cells and confirmed that they were indeed immunosuppressive. Somewhat to our surprise, this did not translate into a measurable clinical effect, so that when we dichotomized patients into high and low Treg counts, there was no effect at the onset of treatment, after the first cycle, or when we actually measured the actual effect, the induction of T cells during regulatory T cells during a cycle. So from this, we conclude tentatively that yes, Tregs are induced, but there is no measurable impact of the induction of T regulatory cells in this trial. What about subset of CD8 positive T cells? Well, I'd like to begin by showing the lineage relationship of memory and effector T cells. And this is a somewhat controversial issue, but still most investigators would adhere to the idea that you start by naive cells that differentiate into T effector memory cells and T effector cells. And what we did in this trial was to sort out all of these phenotypes to determine their potential impact on outcome. And what we saw was that on the left-hand side of this slide, we saw that there was not much changed in the naive T cells or in the central memory T cells, whereas we saw, interestingly, that in the non-relapsing patients on the right-hand side of the slide here, there was actually a reduction of T effector memory cells in parallel with an induction of T effector cells. So we postulated that there may be a transition of, transition of T effector memory cells into T effector cells during therapy. And this proved to be the case, and what we did was to analyze whether patients had experienced an induction of T effector cells along with a reduction of T effector memory cells, and these patients are found here. And as you can see, this is where many of the non-relapsing patients accumulate in this trial. If you analyze it more carefully, you will see that patients who experience this transition of effector memory to effector cells fare significantly better than those patients who do not, both in terms of leukemia-free survival and overall survival. We were surprised and somewhat encouraged by the fact that we saw a similar relationship in older patients. So in the cohort of patients above the age of 60, again, patients who had experienced T cell transition, as we call it, during the first cycle of Ceplene IL-2 had much better prognosis than patients who did not. If you want to phrase it superficially, one could say that if a patient experiences transition of T cells during therapy, it doesn't matter if you're young or old. Now, finally, NK cells, we saw, as expected, an accumulation of NK cells during each cycle of Ceplene IL-2. It comprised both the immature subset of CD56 positive, CD16 negative NK cells, which are the precursors to the CD16 positive NK cells. You can see that this is before therapy and after therapy, both the, sorry about this. These are the mature cells clearly accumulating, and these are the more mature cytotoxic cells. And we saw that in essentially every patient with during both cycle 1 and in subsequent cycles. Now, these effects on NK cells were weakly predictive of a leukemia-free survival, which survived in multivariable analysis, but I will instead go into details of NK cell function measured by the expression of natural cytotoxicity receptors. As You know, NK cell function, their cytotoxic function is dictated by an array of inhibitory receptors dominated by the killer immunoglobulin type receptors, the KIRs, and the natural cytotoxicity receptors, in particular in this trial measured as the expression on NK cells of NKp30 and NKp46. NK cells require these two and other activating signals to properly attack and destroy, for instance, leukemic cells. Now, we saw a strong induction of NCRs, both NKp46 and NKp30, during cycles of immunotherapy. And interestingly, we observed that this translated again into significant benefit for the patients. In all patients analyzed, we saw that if you dichotomized patients into a high and a low group, there was a survival advantage in terms of leukemia-free survival and overall survival of patients who had NK cells expressing high numbers of NKp46. This was done after the completion of the first treatment cycle. Again, older patients actually showed the same trend. A patient within the group of older patients, you could see that patients who had high expression of NKp46 showed unexpectedly, I would say, very good survival and leukemia-free survival in this trial. Okay, this is my last slide. I will just show you two mechanisms that are regulated by Ceplene in this trial. NCRs and T-cell transition, are they independent? We did the following experiment. We analyzed patients who didn't have transition and didn't have functional NK cells. Very poor prognosis. If you had only one, not significantly better prognosis, but you seem to have benefit of both experiencing transition and having functional NK cells, and this translates into excellent overall survival. This is my summary. Histamine is a NOX inhibitor that optimizes immune-mediated clearance of leukemic cells. Used in conjunction with low-dose IL-2, it prevents relapse of AML in a phase 3 trial. And there seems to be, with the due reservations, pronounced efficacy in monocytic AML. And finally, during therapy, NK cells and subsets of T cells are activated that may prognosticate outcome. We would like to pursue these studies into further investigation with your help and others, and are planning for a phase 3 trial that will comprise validation of these findings. Thank you very much.
Thank you very much, Christopher, for these really interesting results. Are there questions? Just one question. You made a relation to the FAB subtype. Did you also look for molecular subtypes?
Again, the FAB subtypes?
The response to Ceplene, did you look for molecular subtypes of AML?
Yes. I mean, you run into small numbers rather rapidly when you do this. If you look into the phase 3 trial, there's no, the treatment benefit does not look to be dominated by any particular molecular marker. So, for instance, karyotype does not seem to play a major role. The trial was independently significant, for instance, within the group of normal karyotypes. So I think this is rather a generic effect, as I would expect from an immunotherapy.
It would be nice to see, I mean, you had this beneficial effect also in the older patient population, which usually have an unfavorable karyotype. Yes. So it would be nice to see whether this is a group that also benefits. Are there other questions? Why is the drug not more frequently?
No, there was one question up there, I think.
Who is? Oh, sorry.
The toxicity of histamine that you add to your IL-2?
Well, histamine produces transient side effects in patients, comprising flushing and tachycardia, for instance. It lasts only for approximately 1 hour. There are no, as we know it, long-term effects of administering histamine to patients. So it's a relatively non-toxic agent. As I was emphasizing, patients, you cannot measure deterioration of quality of life during therapy. So it's not a very toxic agent.